Tansky, and I will be showing you cell-mediated immune response.
The pathogen enters the body and encounters an antigen-presenting cell, which in this case is a dendritic cell. The dendritic cell engulfs and degrades the pathogen and forms this into antigen fragments. These antigen fragments are taken up by the class II MHC molecule. Then the specific helper T cell will bind to the antigen fragment on the surface of the dendritic cell. The antigen and the MHC molecule bind specifically to the variable region of the antigen receptor, or specific amino acid sequences. The constant or C region makes up the rest of the antigen receptor, which is constant throughout all T cell receptors. An accessory protein called the CD4 also helps with binding of the helper T cells and the MHC molecule. The binding and activation of the helper T cells causes it to secrete chemokines, which causes clonal selection or gives rise to a clone of helper T cells and cytotoxic T cells.
First, the newly made cytotoxic T cell travels through the bloodstream and matches to an infected cell with a corresponding antigen to the cytotoxic T cell antigen receptor. The cytotoxic T cell will bind with a class I MHC molecule and an accessory protein, CD8. Once they are bound, the T cell will release perforin molecules, which will form pores on the infected cell. The T cells will also release granzymes that will go through the pores created on the infected cell by endocytosis. Once inside, the granzymes will initiate apoptosis, causing fragmentation of the nucleus and eventual death. The cytotoxic T cell then leaves the dying infected cell and goes on to attack other infected cells. Meanwhile, the cloned helper T cell goes off to make memory helper T cells that remember the antigen for future use. If a second exposure occurs, these memory cells can immediately create active cytotoxic T cells to kill the infected cells during this second response.